There's this very specific, almost universal experience of just standing in front of an open medicine cabinet at like 2 in the morning.
Oh, absolutely.
Now, you've got a headache, maybe a slight fever, and you're just staring at the shelves. On the left, there's a daily prescription you've been taking for months. And on the right, an over-the-counter pill you just picked up.
Right.
And for a split second, as you hold a pill in each hand, you wonder what actually happens when these little chemical capsules meet inside my stomach.
It's a profoundly valid fear, honestly. Though we rarely vocalize it, we're sort of conditioned to view medicines in total isolation,
Yeah, like they're just doing their own thing.
Exactly. We think of them as these tiny, single-minded workers that line up neatly, go to their assigned organs, do their jobs, and leave. But, the biological reality is far more chaotic.
Chaos is a good word for it.
When you mix medications, you're entering the really volatile territory of drug interactions, or DDIs. It's arguably the most pervasive, invisible danger of modern medicine today.
And that invisible danger is exactly what we're hunting today. In this deep dive, we are going to thoroughly decode how artificial intelligence and, machine learning are revolutionizing our ability to predict these sometimes fatal chemical clashes long before a drug even reaches your pharmacy.
It's a huge shift in the field.
It really is. We're working through a massive stack of research today, diving into the sources to map this incredible transition from computational discovery in a lab to actual practical clinical application.
Well, the best place to start is by meticulously defining the battlefield here. When we say DDI, we are talking about a pharmacological or clinical modification that's induced by the co-administration of multiple drugs.
Right, taking them at the same time.
Yes. And the medical field traditionally divides these interactions into 3 distinct categories. First, you have pharmaceutical, or PC.
Which is what exactly?
Those are chemical reactions that happen before the drug even enters the body, like mixing 2 incompatible liquids in an IV bag, and they instantly crystallize.
Oh, wow. Okay, so outside the body entirely.
Exactly. Then second, you have pharmacokinetic, or PK, and third, pharmacodynamic, or PD.
We'll get into both of those heavily today.
We will, because in complex diseases like cancer or cardiovascular disorders, prescribing combination therapies is the absolute standard of care. But that concurrent use exponentially increases the risk of an adverse DDI.
Okay, let's unpack this. Because for decades, the way we caught these interactions felt painfully slow and, honestly, really reactive.
Extremely reactive.
We largely relied on phase of 4 trials, post-marketing surveillance, or, limited in vitro and in vivo studies in the lab. Basically, we were either waiting for a patient to get sick after a drug was released.
Which is the worst case scenario.
Right. Or we were running incredibly expensive, isolated lab tests. But given the sheer volume of novel synthetic therapeutics we have today, exhaustively testing every infinite combination on human subjects isn't just a financial nightmare, it's mathematically impossible.
The combinatorial explosion is staggering. You just cannot physically test 1 million chemical pairings in human trials.
So we need AI.
We do. But before we can even begin to understand how an artificial intelligence predicts these clashes, we really have to understand the physical reality of how drugs collide in the human body.
Right. The biology.
Exactly. We need to look closely at that second category. Pharmacokinetics, or PK. Simply put, PK is the study of the movement of drugs within the body, and it's governed by a sequence of 4 stages, universally known as the ADME journey.
ADE.
Yes. That stands for absorption, distribution, metabolism, and excretion.
I always find it helpful to visualize things. So as we walk through this, I want you to imagine the human body as a massive, continuous chemical processing plant.
That's a great analogy.
Absorption is the loading dock, right? Usually the stomach or intestines where raw materials are taken in. Distribution is the conveyor belt network carrying supplies everywhere via the blood.
Correct.
Metabolism is the manufacturing floor, mostly located in the liver, where enzymes break things down. And excretion is the waste management system, primarily the filters in the kidneys. So let's look at a collision right at the loading dock. What happens there?
Well, the loading dock is highly sensitive to the surrounding environment, particularly acidity. Consider the collision between a basic over-the-counter antacid and a powerful antifungal medication called ketoconazole.
Okay, an antacid and an antifungal.
Right. Ketoconazole is highly dependent on a severely acidic environment just to dissolve. It physically requires a gastric pH of less than 3 to break down into a form the body can actually absorb.
But antacids, by their very definition, neutralize stomach acid.
Yeah.
They push the pH up.
Exactly. They raise the pH to greater than 4. So you swallow the ketoconazole capsule, it sits in the stomach, and it simply cannot dissolve. It creates a phenomenon known as dissolution-limited absorption.
So the physical breakdown just stops.
It stops completely. The clinical result is a staggering drop up to 66% in the drug's concentration in the bloodstream. The fungal medication just passes through the body as an inert, undissolved rock.
Wow. So the loading dock is flooded and the cargo just washes away. That's wild. Let's move deeper into the plant then, down to the manufacturing floor metabolism. The liver is packed with enzymes. Is it easy to cause a traffic jam there?
It is remarkably easy and often incredibly dangerous. Let's look at a common antibiotic called clarithromycin colliding with statins.
Statins are standard cholesterol-lowering medications, right?
Very common, yes. So clarithromycin acts as a powerful inhibitor. It aggressively binds to a specific crucial recycling enzyme in the liver called CYP3A4, as well as the OATP1B1 and OATP1B3 transporters on the membrane of the liver cells.
So in the context of our factory analogy, the antibiotic basically padlocks the manufacturing floor doors and breaks the recycling machine.
It shuts the whole system down. Because those transporters are blocked, the liver cannot take up the statins to process them.
Wait, so the statins are just stuck?
Exactly. Because the statins cannot enter the liver, they have nowhere to go, so their plasma levels in the bloodstream skyrocket.
That sounds bad.
It's very bad. This elevated toxicity risks severe acute kidney injury, myopathy, and rhabdomyolysis, which is a condition where your muscle tissue rapidly breaks down and floods the bloodstream with toxic proteins.
All because an antibiotic closed the door on a cholesterol med. That is terrifying.
It highlights why this is so critical.
So if a drug somehow survives the stomach and gets past the liver, it eventually has to leave the body. Let's look at the waste filters. Excretion. I was reading in the sources that a clash here isn't always a bad thing.
It's a fascinating exception to the rule, actually. Take probenecid, which is a gout medication, and penicillin. Both of these drugs want to exit the body through the exact same route.
Which is where?
The organic anion transport system in the renal tubules of the kidneys. But probenecid is highly competitive. It effectively fights for that active transport mechanism and blocks penicillin's exit.
So it barricades the waste door, trapping the penicillin inside the building.
It forces it to stay in circulation, yes. This prolongs the penicillin's half-life in the body. It's a rare, brilliant example of a beneficial pharmacokinetic interaction that doctors use therapeutically.
Oh, I see. Like when they want to keep an antibiotic in the patient's system for a longer duration without having to increase the actual dose.
Precisely.
Okay, we've covered a lot of dense biology here, so let me just wrap my head around this first leg of the journey. We'll do a quick 5-minute recap.
Great idea.
We've established that testing every drug combination is mathematically impossible, which is why AI is so desperately needed. But first, we mapped the ADME journey absorption, distribution, metabolism, and excretion. We saw antacids neutralizing acid and stopping ketoconazole dead at the loading dock. We saw clarithromycin blocking liver transporters and causing massive statin toxicity. And we saw probenecid intentionally blocking the renal exit to boost penicillin.
A perfect summary.
It really makes you look at your own medicine cabinet differently, doesn't it? Like, think about the medications you or your family take. Have you ever considered that a simple antacid could completely neutralize a life-saving prescription just by changing the pH of your stomach?
It's a sobering biological reality, and you have to keep in mind that is solely pharmacokinetics, the mechanics of where the drugs travel and how they survive the journey. Now we must transition to the next phase, pharmacodynamics, or PD.
Right, the third category.
This is the study of what the drugs actually do to the body's receptors once they finally arrive at their destination.
Oh, I like this. So if PK is the delivery truck navigating the highway, PD is the actual installation of the furniture inside the house.
The installation process relies heavily on receptors, which you can think of as biological locks waiting for a chemical key. Pharmacodynamics explains how drugs bind to these receptors, or inhibit enzymes, to produce a biological effect.
So they're physically interacting with the cells.
Yes. And when two drugs interact at the PD level, they generally fall into two broad categories. Synergistic, where they enhance each other's effects, or antagonistic, where they block each other.
And synergy can be a lifesaver, right? The sources talk heavily about aspirin mixed with P2Y12 inhibitors, like clopidogrel.
This combination forms the absolute foundation of standard dual antiplatelet therapy, or DAPT. It's typically given after heart attacks or stent placements. Both drugs target entirely different biological pathways to achieve the exact same goal.
Which is stopping blood platelets from clumping together and forming
Yeah.
clots.
Exactly. Their synergistic effect is incredibly powerful. But there's a catch. Long-term use drastically increases severe bleeding risks.
Right, because the blood won't clot at all.
So to manage this, modern medicine uses genetic or platelet function testing to guide a de-escalation strategy. Doctors meticulously lower the dose of the P2Y12 inhibitor over time to perfectly balance the antithrombotic efficacy with the patient's bleeding risks.
So they're working together to save the patient. On the flip side, we have competitive antagonism, where drugs are actively fighting for the same chair. The most famous example here has to be naloxone and morphine.
A classic and vital example. Morphine binds to opioid receptors in the brain to produce profound pain relief, but in excess, it suppresses the central nervous system, causing fatal respiratory depression.
People just stop breathing.
Naloxone is a lifesaver because it has a significantly stronger chemical affinity for those exact same opioid receptors. However, unlike morphine, naloxone does not activate the receptor.
It just sits there.
It acts as a hyperaggressive blocker. It physically displaces the morphine, instantly reversing the opiate effects and restoring breathing.
Now, here's where it gets really interesting in the sources. Dual effect interactions. What happens when two drugs do the same job, but take completely different paths to get there, like taking beta blockers and calcium channel blockers together?
Both are very commonly prescribed to lower blood pressure, but through entirely different mechanisms. Beta blockers target beta-adrenergic receptors to slow the electrical signal of the heart, reducing its rate and contractility.
And the calcium channel blockers.
Or CCBs, they inhibit L-type calcium channels in the smooth muscle cells, causing vasodilation, which is the widening of the blood vessels.
That sounds like a great 1-2 punch. Slow the pump, widen the pipes.
In theory, yes, but in practice, the synergistic effect on the cardiovascular system can be overwhelmingly powerful. Combining them can cause severe bradycardia.
A dangerously slow heart rate.
Right, or even a complete heart block where the electrical signals fail entirely. It's a dual effect that requires extreme caution and monitoring. We see a similar complementary danger with diuretics and ACE inhibitors. Diuretics target the kidneys to excrete sodium and water, reducing overall blood volume. ACE inhibitors block the angiotensin II enzyme to widen blood vessels. Together, they aggressively drop blood pressure. But in a patient who already has underlying renal impairment, starving the kidney of that blood flow can trigger acute kidney injury.
Man, the body is so fragile. Now, the sources also mention pharmaceutical synergy, which is a bit different. This is when drugs team up to destroy an outside target, like carbenicillin and gentamicin taking on tough bacteria.
Carbenicillin is a beta-lactam antibiotic. Its specific job is to destroy the structural cell walls of bacteria. Gentamicin is an aminoglycoside that slips inside and attacks the bacteria's 30S ribosome, completely stopping protein synthesis.
So one breaks down the wall, the other poisons the inside.
Exactly. Their mechanisms don't conflict at all, so together they synergistically eradicate incredibly tough resistant bacteria like Pseudomonas aeruginosa.
Okay, so far we've talked about drugs hitting other drugs, but one of the most surprising things in this research is that sometimes the interaction isn't another pill, it's what you had for breakfast. We have to talk about grapefruit juice.
Ah, yes. Grapefruit juice is a fascinating anomaly in pharmacology. It is remarkably dangerous when combined with certain medications.
I always thought that was just a myth.
Not at all. It contains complex chemical compounds called furanocoumarins. These compounds cause what the field calls mechanism-based inhibition in the intestinal tract. They don't just temporarily block the CYP3A4 digestive enzyme like a traffic jam. They irreversibly bind to it and destroy it.
Wait, they physically destroy the enzyme?
The enzyme is permanently inactivated and degraded. Because the body can no longer metabolize the drug during the initial absorption phase, the drug's oral bioavailability, the amount entering the blood, shoots up to highly toxic levels.
That is insane.
And this effect doesn't just fade in a few hours. It lasts until your body physically transcribes and synthesizes brand new enzymes from scratch.
Which takes how long?
It can take up to 72 hours.
So a glass of juice on Monday morning is still altering your body chemistry on Wednesday?
Yes, it is fundamentally altering your metabolic baseline. Contrast this with direct pharmacological contraindications, like taking MAOIs, an older class of antidepressant, with sympathomimetic amines, which are found in certain cold medicines.
That's what happens here.
That specific combination causes a massive, unregulated release of norepinephrine in the body, leading to a sudden, potentially fatal hypertensive crisis. It is an absolute contraindication.
All right, let's pause again and digest that second leg of the journey for our 10-minute recap.
Let's do it.
We shifted from the delivery routes of PK to the lock and key mechanisms of PD. We covered synergistic therapies like aspirin and clopidogrel, the aggressive antagonism of naloxone, and the dangerous dual effects of combining blood pressure meds that slow the heart while widen the pipes. And we just learned that grapefruit juice literally chemically destroys your intestinal enzymes.
It's a lot to take in.
So here is the overarching problem for you to think about. If a simple breakfast juice can permanently alter your enzymes, imagine the sheer complexity of predicting chemical clashes across thousands of novel synthetic drugs interacting with diverse human biology. How can we possibly calculate all that?
And this is exactly where the core of our deep dive begins. The biological chaos we just mapped out requires a massive computational pipeline to understand. Artificial intelligence and machine learning are stepping in to solve this combinatorial explosion.
Finally, the AI heroes arrive.
The standard ML workflow for DDI prediction involves 4 meticulous stages: data collection, model construction, evaluation, and experimental verification.
The AI basically has to learn everything we just talked about, but for every chemical ever discovered, where does it even get that education?
The data acquisition stage pulls from massive, highly specialized global repositories. The AI reads databases like DrugBank to map drug targets, PubChem to understand 3D chemical structures and bioactivity, KedG to map intricate human metabolic pathways.
It's just devouring data.
Entirely. And databases like 2Size and PharmGKB to ingest decades of known clinical side effects and genetic variants.
Yeah, but how does the AI actually see the drug once it has that data? This is the representation level in the sources.
Oh, I get it. It's like trying to figure out if 2 people will get into a fight at a party.
Go on.
You have 2 different ways to evaluate them. You can look at their shared hobbies and the friend groups they hang out with, which is similarity. Or you can sequence their actual DNA to look for aggressive traits, which is structure.
That is a highly effective analogy. In similarity-based methods, the algorithm operates on the assumption that drugs with similar profiles will interact in similar ways. A standout model in the research uses support vector machines, or SVM.
How does that work?
It calculates a mathematical pairwise kernel across 5 different similarity metrics simultaneously. It looks at 2D molecular similarity, 3D pharmacophoric similarity, interaction profile fingerprints, target similarity, and adverse drug effect similarity. And the research said it achieved a 0.97 AUROC. Let's translate that for me. If a perfect score is 1.00, what does 0.97 actually mean in the real world?
Think of it this way. If you give this AI 1 pair of drugs that definitely interact and 1 pair that definitely don't, the AI will correctly identify the dangerous pair 97% of the time. It is an incredibly reliable metric of predictive accuracy.
That's similarity. What about the DNA method, reading the drug itself?
That is structural representation. The AI bypasses the side effects and reads the architectural blueprint of the molecule. It looks at raw molecular graphs in something called canonical SMILES.
SMILES?
Yes, the Simplified Molecular Input Line Entry System. SMILES is a revolutionary protocol that translates a complex 3D chemical structure into a 1D string of text characters, allowing a computer to read a molecule literally like a sentence.
So once the AI has this data, either looking at its friends or reading its text string, how does it actually learn? The sources broke down 2 learning paradigms: multitask learning versus traditional classifiers.
Multitask learning recognizes that drug interaction data is often incredibly sparse and fragmented. A standout here is Zhu et al.'s BIL-STM network. It is designed to extract DDI information hidden in millions of pages of textual biomedical literature.
It just reads the medical journals.
Essentially, yes. It represents sentences as word embeddings combined with position embeddings. By jointly learning 2 tasks, simultaneously predicting if an interaction exists, and classifying the type of interaction, it avoids the trap of focusing too narrowly.
Oh, well, that makes sense.
Another brilliant model is DEMIL, which optimizes 5 synergy regression tasks and a classification task all at once. This avoids the seesaw effect.
The seesaw effect.
Yes, where improving the AI's accuracy on one specific metric accidentally harms its accuracy on another.
Got it. What about traditional classifiers? Do they still have a place?
They do, especially when using handcrafted features. A fascinating example is the LCMDS model. It tackles the massive computational load of mapping DDI networks by using the Dempster-Shafer theory of evidence.
That sounds intense. What is that?
It's a mathematical framework that fuses the outputs of multiple local classifiers. It essentially takes differing opinions from smaller algorithms and combines them to handle uncertainty highly efficiently, creating a unified, highly accurate prediction.
Okay, 15-minute recap time. We've mapped how the AI gets its data from places like PubChem, and we've broken down how it sees a drug either by its similarity, the company it keeps, or its structural blueprint.
Yeah.
We also looked at how multitask learning balances multiple jobs to avoid the seesaw effect.
A lot of ground covered.
What's fascinating here is how the AI is literally learning to read a chemical structure like a foreign language.
Yep.
But as these models evolve, the field seems to be moving away from choosing between similarity or structure.
Exactly. The answer the field is arriving at is both. And they are fusing them using the absolute bleeding edge of deep learning, specifically graph neural networks, or GNNs.
We have to slow down here because this is where the math gets intense. A graph in computer science isn't a bar chart, right?
Right. Think of a graph as a massive, multidimensional web. The atoms of a drug are the nodes or connection points, and the chemical bonds between them are the edges or lines connecting them. GNNs treat drugs as interconnected systems.
And the sources mentioned the SMILE GNN model here.
Yes, take the SMILE GNN model. It perfectly fuses the two representations we just discussed. It integrates those 1D SMILE's text features with the complex topological features of a knowledge graph.
And then there is the ACD-GN model, which the research said eliminates entity heterogeneity. Entity heterogeneity sounds like a very polite way of saying apples and oranges.
That is a perfect translation. When you feed an AI biological data, you are giving it chemical structures, genetic sequences, and clinical symptoms. These are fundamentally different domains.
They don't naturally speak to each other.
Entity heterogeneity is the mathematical friction of trying to compare them. The ACD-GNN uses an attention-based cross-domain network to act as a universal translator, seamlessly aligning all these different data types so the AI can find hidden connections between a gene mutation and a chemical bond.
And some of these models don't even need humans to build them anymore, right? The auto-DDI model uses reinforcement learning to automatically design its own optimal GNN architecture for whatever dataset you give it, adapting the depth of its layers on the fly.
It's remarkably autonomous.
And beyond graphs, there's matrix factorization, like the GRPMF model. Reading about that felt like watching an AI play a massive game of Sudoku, filling in the blank spaces of a drug interaction matrix using graph-based regularization.
To handle all this simultaneous math, the AI needs intense focus. That brings in attention mechanisms.
Think of attention mechanisms, like in the MABL model, as a spotlight in a pitch-black room. Instead of looking at every single atom of a massive drug molecule equally, the AI dynamically focuses the spotlight only on the specific drug attributes or substructures that actually trigger an interaction, completely ignoring the background noise.
And then we have Siamese networks like CNN-SIAM. This utilizes contrastive learning.
Siamese, like twins.
Imagine twin, identical convolutional neural networks. They process multimodal data for two different drugs simultaneously, side by side. They compare their internal weights in real time to predict if the pair will interact.
Finally, we can't talk about AI right now without talking about the generative AI revolution and large language models.
Indeed. LLMs are changing pharmacovigilance. The LLM DDI model uses GPT embeddings to capture incredibly diverse molecular information from biological knowledge graphs, refining that data through a GNN framework.
Wow.
Another model, DDI-GE, uses in-context learning. It retrieves similar historical examples and uses a GPT-4 ensemble discriminator to evaluate zero-shot and few-shot biomedical data. It essentially acts as a panel of expert digital judges reviewing a case.
So we have built the ultimate predictive machine. It can read a chemical structure, map it to the human genome, translate apples to oranges, and predict a fatal clash with near-perfect accuracy. But bringing this from the computational lab into an actual hospital bedside comes with a massive hurdle: the black box problem. Because if this AI predicts a fatal interaction, but the doctor asks why, the AI can't just say because my math says so.
The interpretability crisis is the final frontier. In clinical practice, an AI prediction without a transparent explanation is functionally useless. A doctor will not alter a life-saving treatment plan on blind faith.
Well, of course not.
This is driving the rapid development of explanatory AI. We now have tools like GNN Explainer, which visually highlights the specific molecular subgraphs that influence the AI's decision.
So it literally points to the exact atom on the screen and says, this right here is the problem.
Yes. Another powerful model, SHAPGNN, uses game theory, specifically Shapley values, to quantify exactly how much each individual molecular feature contributed to the final prediction, dividing up the credit for the interaction.
Show them the math, basically.
And the DDI model uses a dual-stage attention mechanism with Bayesian calibration to capture temporal dependencies. It significantly reduces false positives and shows the doctor the underlying statistical logic.
But even with crystal clear explanations, real-world translation is a bureaucratic and logistical mountain. Integrating multimodal data, meaning successfully combining a patient's real-time genome sequence, the molecular drug data, and their current clinical lab tests, requires massive cross-domain integration across siloed hospital systems.
And it must all comply with incredibly strict regulatory frameworks. Algorithms must navigate the EU AI Act and the FDA's Software as a Medical Device, or SaMD, guidelines. Data privacy, patient consent, and cybersecurity are paramount when you are processing this level of personalized biological data.
So what does this all mean for you, the listener? It means these machine learning models are no longer just computational novelties running simulations in a university basement. They are actively reshaping pharmacovigilance and precision therapeutics. They are looking into the murky, chaotic depths of human biology and saving lives by seeing the invisible clashes before they happen.
The transition from exhaustive, reactive human testing to proactive, predictive modeling is perhaps the most significant pharmacological leap of our generation.
As AI gets better at predicting these interactions, we might soon reach a point where your exact genetic profile and your daily diet are instantly cross-referenced against millions of chemical combinations before a doctor even writes a prescription. But who should make the final call on a high-risk medication? The human doctor relying on decades of bedside experience, or the algorithm that has read every medical paper ever published? Think about that next time you open your medicine cabinet.